All right, it is 2 o'clock Eastern. I am Matt Zeman. I'm the Learning Chair here from EO Raleigh-Durham, and welcome to our 8th virtual learning event out of our series that we are calling Coronavirus and Your Business. And we have an amazing session for you today and some wonderful things coming each Thursday at 2 o'clock in the coming weeks. Amy Henning is sitting in our panel today. She's our chapter manager. She's been amazing putting these events together. And then also Olivia at EO Global. Thank you so much for helping us get the word out. Really appreciate the people, the attendance on these events, and hopefully we're providing good content for you. So what started as a way to kind of connect with just our chapter in light of canceling our in-person events in the future has turned out to have a lot more interest. So we'd like this panel today to be as interactive as possible. And we're going to plan to open up the conversation towards the end of this so that you can ask questions and share from your own experience. To do that, there's two different ways. You can either raise your hand, which lets us know that you want to be called on to actually ask the question to the panelists yourselves, or you can write your question down in the Q&A panel. And by doing that or voting on questions that are in the Q&A panel, we will know to read those questions for you and to have our panel respond. Before we get going, just a couple other things. Next week, Thursday at 2 o'clock, there's a program being done by Culture Index on how to apply behavioral analytics to maximize your the productivity of your employees. And they're doing, if you're an EO member and you're on the call, if you go to our website at eoraleigh.com/coronavirus, you'll see their offer for a free, where basically they'll test your whole team for you and then give you as the CEO EO member a 90-minute consultation of the results, all for free. So that's Culture Index. It's a national company, so anyone in the States can take advantage of that. And again, it's for EO members. I know there's some non-EO members on the call today. The following week, a number of you have probably maybe even come to Chapel Hill to do one of the Dr. Bell programs. He is going to do a program on how to get yourself, your people, and your business starting to sprint again. So always a good session with Dr. Bell, and that's coming up on May 7th. Today's topic is how we will science our way out of the coronavirus pandemic, and it's led by EO Raleigh member Giles Shih, Dr. Giles Shih. Giles has two other experts with him. I'm going to just turn it over to him to introduce the experts. If you have other ideas for content, you'll see that in the chat panel, my email. Please send me your ideas. But with all of that, Giles, why don't you take this away, introduce your panel, and I'm going to be quiet.
Very good. Thank you, Matt, and thanks for organizing this. Thank you for joining us today. My name is Giles Shih, and like Matt said, I do have a PhD in microbiology and have studied viruses and bacteria, but I'm no means a coronavirus expert. But I've gathered two experts with me today to serve on the panel, and you'll be hearing from them in a few minutes. We've got first Deepa Maka. She's a board-certified physician for emergency medicine, and she's a member of EO Detroit, and she's joining us today. And she's going to be covering the medical aspects of coronavirus and COVID-19. And then also after that, Jeff Banks, CEO of Nuventra here in the Research Triangle, and a member of our EO Raleigh chapter, is going to cover the drugs and vaccines that are under development to combat COVID-19. There's a lot of information and a lot of stuff out there. It's being updated daily, and hopefully we can provide some useful information to you as we start this dialogue and tackle this very critical issue facing us. So before we get started, you might be scratching your head about the title. It's not very grammatical, I know, because it's you know, science is a noun, it's not a verb. But Matt, if you advance to the next slide, I was inspired by this quote from the movie The Martian, which was originally a book. You might have seen it, Ridley Scott movie, and the character in there, played by Matt Damon, he's finds himself stranded on Mars by himself with limited food and water, and he realizes, I'm going to have to science the blank out of this. And I feel like that it speaks to the kind of the spirit and ethos of our times that we find ourselves in. using science to figure out these tough problems that we're faced with by combining cumulatively our knowledge, our data, and facts, and trying to solve for this. And this is the situation that we find ourselves in with the coronavirus pandemic. And in this day and age where we have countries battling each other for PPEs, protective gear, and people casting blame on each other for who started the virus, it's really refreshing to see the scientific and medical communities really come together all across the world and collaborate in an unprecedented way to figure out how to control this virus, come up with vaccines, come up with drugs, come up with just ways to fight this global pandemic. So I want to, the take-home message I want to give today is a message of hope that together with the scientific and medical communities working collaboratively around the world, we can get this coronavirus COVID-19 pandemic under control. But it's going to take time, and it's going to take all of us working together and doing our part. So I'm going to start off by talking a little bit about what we know about COVID-19 and the coronavirus, where it came from, and where it looks like it will be headed. And then Deepa's going to, like I said, talk about the medical perspective, and then Jeff will come in with a message about how drugs and vaccines which ones are under development, and what the timelines look like to fight this virus. Okay, so next slide. So what is COVID-19? COVID-19, and you might have heard coronavirus, and it's kind of used interchangeably in the media. Basically, COVID-19 is the disease that's caused by this novel coronavirus, and the scientific name is SARS-CoV-2. It's a novel coronavirus, and basically a coronavirus, just like the name implies, is this basically a sphere or bubble of that contains this DNA or genetic material. In this case, it's RNA, and it's has this crown of spikes on it, the proteins that allow it to bind to the cells and infect human cells. And once it binds to it, it injects the RNA into the cells and then propagates more copies of the virus. And so of the coronaviruses that are known, there's over 200 types that are known. Only 7 of them affect humans. 4 of them are responsible for the typical common cold that you see. About 15 to 30% of common colds are caused by these 4 types of coronaviruses. There's 2 types that have been involved with epidemics in the past. You might have heard of SARS back in 2002, hit Hong Kong and parts of Asia very hard, and MERS hit the Middle East back in 2012. Now, this new novel virus, SARS-CoV-2, or what we're also referred to as COVID-19, the causative agent of COVID-19, is what's called a pandemic strain. It's the past 2 types were SARS and MERS were more epidemics. They didn't reach the pandemic status. This has spread globally to all countries and continents throughout the world. And this virus is the one that is most of interest right now and being studied by all the medical and scientific communities. Next slide. So, like I said, it's likely that the coronavirus, as most coronaviruses, are resident in animal species, and this novel coronavirus somehow was able to jump from animals to humans, and it evolved this ability to infect humans. And because humans had never seen this particular novel virus before, it was able to spread widely, first from Asia and China to Europe, and then now North America and all over the world. And a key difference, and we'll go into more of the details of how it spread later, but the key difference is that in terms of COVID-19 or to SARS in the past, is that there has been documented cases of pre-symptomatic transmission. In other words, people that don't have any symptoms can spread the virus, can shed the virus for a number of days. And that's what has allowed this virus to continue to spread very rapidly because, for example, in cases like SARS, or you also may have heard of Ebola, is very deadly diseases. People get very sick very fast and get hospitalized, and so they aren't out and about in the community continue to spread the virus. It's mostly those that are in the immediate family or in the healthcare facilities that do get exposed to those viruses. But this is unique in that there's an incubation time of anywhere from 4 to 5 to 7, sometimes even 2 weeks potential of the time in which you are not symptomatic but could be shedding the virus. So that's what we know so far. And then this is a slide that is updated. There's a screenshot from a website that's updated on a regular basis. This is, I took this screenshot on Tuesday, so that number has probably even changed from then. But you can see there's 2.5 million cases all over the world, 170,000 plus deaths. And it's on every continent except for Antarctica. So it's everywhere. And it's rapidly spreading and also very deadly because people have not don't have any immunity to this. It's not like, the flu virus comes around every year, and because people have been exposed to and have flu, have recovered, they have some antibodies and some resistance, immunity to it, and so it's not as lethal or widely spread. There's also herd immunity that prevents the further spread of the flu. But this is new. This is a novel coronavirus, and this is why it's been able to spread so quickly and be so deadly. Next slide. So I wanted to talk a bit about and look at kind of the epidemiology of how the disease is spread. And this site, again, is updated very regularly. And I took this screenshot on Tuesday, showing, and this is a logarithmic scale. So you can see that in the early days, it has an exponential curve that one person affects 2, 2 affects 4, and 16, and so on and so forth. And so as you plot this, essentially in the early days of the virus, it was doubling, cases were doubling every 3 days. As we have gone through this process over the last several weeks, we can see that curve is quote unquote flattening. It's starting to level off. There's still cases that arise, but you see it's fallen from the exponential phase and it's starting to flatten out. That blue line is the United States, which now has the most cases so far in the world. And this website tracks against all the other countries. And you can, if you are interested, to go in and you can look at all the different countries and the numbers. But the point being is that that curve is starting to flatten. There's still cases, but because of the measures we've taken with social distancing and the precautions that we've taken, the cases have started to increase slower. They haven't decreased yet, but they're starting to flatten out. And the next slide shows North Carolina, because many of us are joining this webinar from North Carolina. I wanted to show how North Carolina compares to other states across the country. North Carolina put a stay-at-home order fairly early at the end of March, so they were fairly proactive, and you can see that as a result of that, the social distancing and the precautions put in place, and maybe also the density isn't as great as, say, in New York. We were able to flatten that curve earlier and sooner. And in fact, I think later this afternoon, the governor of North Carolina is going to be making a press conference talking about how we plan to start to open up the state for business. And this will be a key issue and question and challenge as we go forward, how to go back to work, return to work, or return to life as we know it without causing more cases to spike up. And the key is to, as we've all heard, to flatten that curve so we don't overwhelm the healthcare system and to be able to trace those that do have the illness. Okay, next slide. So what does the future look like? First, the good news is that, as I've shared in the past 2 graphs, the curve is starting to flatten. The precautions that we've taken, the social distancing, the stay at home, it is having an impact, and the cases, while they're still increasing, they're increasing at a slower rate. So a key question I'm sure a lot of you are thinking about, myself as well as a small business owner, how does the return to work scenario look like? And that's something that will be worked out probably on a week to week, state by state basis, but it will include some of the factors like social distancing and wearing of masks that we've seen and heard about in the media, and we'll cover some of that later. There'll be testing. Testing is such an important part of this, both the testing for the live virus, as well as testing serologically to see if you have immunity to the virus. And that's going to be a key part of this going forward. And then contact tracing, as I mentioned, that's just detective work. You know, whoever gets sick is positive. Doctors will need to come around and figure out what other people they've been in contact with and quarantine those to prevent the further spread. And countries that have done that well have been able to do a very good job to contain the spread of the virus. And then, you know, there's going to be a need to manage the caseload so that we don't overwhelm the healthcare system like what we saw in New York. The better we do these things in terms of social distancing and testing, the better chance we have of keeping the caseload manageable, so doctors can treat and heal those that come down with COVID-19. Long term, there will be a need for a vaccine or a therapeutic, and there will be one developed, but it'll take some time. And Jeff will talk about some of the progress on that. It's a lot of encouraging data, but still, you know, a work in progress. Okay, next slide. So this is my last slide, and just to remind people to check the CDC website for good fact-based guidelines as we look at a return to work environment and scenario, and you know, best practices in terms of frequent hand washing, social distancing, wearing a mask when you're outside to not only protect yourself but also protect others. If you are pre-symptomatic, it just reduces the chance that you'll spread those droplets to others. So those are my comments, and so now I'll turn it over to Deepa, and she'll give us the view from the medical perspective. Thank you.
Hi, everybody. Thank you for having me. I'm Deepa Maka. I'm a board-certified physician in emergency medicine and internal medicine. And I'm also the medical director and owner of Cosmetic Laser MD in Ann Arbor, Michigan. Next slide, please. So just to go over some terms, SARS-CoV-2 is the virus. The disease entity in the spectrum is COVID-19. And as Giles said, the incubation period is 14 days. Most commonly, most people present with symptoms at about 4 to 5 days, but it can be as long as 2 weeks. And that's where we have a lot of people who can potentially infect people. We've talked about social distancing. We all know what that is. And social distancing is really talking about physical space, increasing the physical space between people. Quarantine refers to a person who's been exposed and is at risk of developing the disease. And it basically segregates exposed people from the population. Isolation refers specifically to a person who has known disease and separates themselves from others. So that could be isolation in a home or isolation in, you know, from the rest of the community. Next slide, please. So, you know, Giles referred to most of this, so we can go quickly, but R0 is essentially the number, how it's basically a measure of the infectivity, and it measures how many people one single person can infect. Next. So this is a slide that shows how COVID-19, which is the first one in the second row, compares to others. Influenza has a R0 of 1.5, also known as R0. And and you can see that this number, the one thing that this number also does not reflect is the behavior of individuals because of the long incubation period of COVID-19 and that people can be asymptomatic. It has the ability to, and that is why it's so infectious, to spread so easily in the community. Next slide. So this is just another graphic that shows how one person can infect 2 people, and then how that can go on, and how in the course of the incubation period, this can progress. And this is with an R0 of 2, for example, and COVID-19 is somewhere around 2.5 up to 2.9, they believe. Next. So when we talk about transmission, we talk about, with COVID-19, we talk about respiratory transmission as well as contact. So when we talk about respiratory transmission, we're talking about person-to-person spread via droplets, small and large, similar to influenza, when people say airborne, and coughing, sneezing, talking. So we want to at least be at about 6 feet of distance. So social distancing and the wearing of masks prevents respiratory transmission. Next. So there's, you know, at the bottom there's a link, and the reason I included this link as opposed to the CDC or WHO or ANZIG or one of the other many links out there is I really like the graphic that they had for all of the different types of masks and homemade masks, how to make masks, and what is the difference. So advanced masks, it's very important for people to understand, or for frontline healthcare workers who cannot maintain a physical distance. Their N95, KN95 masks also are now approved, and respirators. And these masks essentially protect frontline healthcare workers from aerosolized droplets. General homemade masks include surgical masks as well as cloth masks. One thing I want to let people know is it's very important to store these masks in paper, not plastic bags. So when we talk about brown bagging masks, this is the reason why, because the fomites in the virus can replicate, and in a paper bag, it decreases that. So what masks do is they protect others from respiratory secretions, as well as they, from our own respiratory secretions, as well as providing another level of protection. I find that mask wearing, you know, during training, also like in the OR or wherever, or in a room when I would be with a patient, prevents me from touching my face, and it provides a new level of muscle memory. Cloth masks need to be washed and reused. Disposable masks should be discarded. Next slide. The second type of transmission is contact, and that's when you come into direct contact with the virus with your mucous membrane, so the eyes, the nose, the mouth. Direct contact with a contaminated surface and then touching your eyes, nose, and mouth, that's how you can become infected. And then we talk about decontaminating surfaces. So gloves and hand sanitizer protect against contact transmission. Next. Very important to remember, I see a lot of people wearing gloves when they're out and about. They do not protect you if you then go and touch your face while you're wearing the gloves. Also, realize that the exterior portion of the mask is contaminated. So if you take off your mask, put it down, and then put it back on, you need to really pay attention to that. Next slide. So how do we prevent? We just finished talking about transmission. Now we're going to talk about prevention. Regular cleaning, disinfecting of all of your environmental surfaces with a virucidal product. The CDC does have a link for EPA approved products and it's not just Lysol and Clorox wipes. There are other things out there that do have virucidal properties. Hand hygiene essentially is 2 things: hand sanitizer with at least 60% alcohol, or washing your hands for at least 20 seconds. Do not touch your face. Eyes, nose, mouth, that's where you have the mucous membranes. Another way to prevent is make sure that you isolate sick contacts in your home, and you want to have a separate bedroom and bathroom if possible. When you leave the house, you wear a mask, and when you're in your home, if there's a sick contact, they should be wearing a mask. Decontaminate before you re-enter your home. Next. So you want to create a circle of safety around your home. So those who live in your home essentially are your circle of safety. The core principle here is physical distance, not social distance, from people who are infected people in the community, and surfaces that carry the virus. Everyone in your home should follow these guidelines. Excuse me. Do not let anyone into your home without an essential reason. Next. So we're going to continue with prevention. Delivery of food packages should be dropped off outside of your door. Do not bring any exterior packaging into your house. When you open these, open them outside your house, wear gloves, and disinfect the packages. Use disinfectant spray, wipe down the exterior, and when you have the contents and you bring them in, make sure you disinfect those contents. So if they're packages of food, wipe down the exterior surfaces. Fresh fruits and vegetables, they should be washed in hot soapy water. Any prepared foods that are ready to be eaten should be microwaved. After you process your delivery, remove your gloves, throw out your gloves, wash your hands thoroughly with soap and water for at least 20 seconds. Use disinfectant spray and wipes on the doorknob, doorbell, etc. after delivery is complete. The whole idea is to make sure your home is a safe haven where you can relax. Next slide. So continuing with cleaning and disinfecting your home, you should be cleaning down and disinfecting your home regularly, all shared hand touch surfaces, wipe down all counters, doorknobs, light switches, countertops, faucets, etc. There is a study that shows that the virus can be detectable in droplets in the air for up to 3 hours and up to 4 hours on some metal surfaces, and 2 to 3 days on other surfaces. So you have to be very vigilant and vigilant. You want to wash your clothes frequently and beddings and towels at least once a week. Next. When you leave your home, okay, it should only be for essential activities. And when you're outside, if you can wear disposable gloves, definitely a face mask, hat, long sleeves, and pants. And you want to maintain a social distance of at least 6 feet. So when you leave the house, again, make sure you take hand sanitizer and wipes if possible. Use them often. Don't touch anything you don't need to be touching. Definitely no shared surfaces. When you're in the store, gloves, mask, hand sanitizer, wipes. Wipe down the cart and wear the gloves. When you're wearing the gloves, don't touch your face. Don't touch anything you don't need to. You should be bagging your own groceries and use credit cards, no more cash. Next. When you come back into your house, now you have to think about decontamination. Remove your shoes outside, spray them with a disinfectant, leave them outside, don't bring your shoes inside. In Asian families, we always leave our shoes outside, so it's a good practice for everybody. If possible, remove your clothes, especially the outer layer. When I come home from the hospital, I kind of derobe and take everything off, and then I go straight into the shower. Use disinfectant wipes on your cell phone, keys, credit cards, wallets, everything that you've used during the trip. I usually start that process in my car. When you come back in, wash your hands with soap and water for at least 20 seconds and keep doing it regularly before preparing and eating meals. Next. So now, what do you do if you get sick? We've talked about transmission, we've talked about prevention. Now we're going to talk about what happens if you did everything you could and then you get sick. Let's make a plan if someone gets sick. Most people experience mild symptoms. If someone gets sick, You should determine in your home where they can self-isolate. Determine who's going to take care of that person and contact that sick person's healthcare provider. Often you can have access to telehealth these days because most providers, most physicians are working in telemedicine. If the sick person becomes ill and has trouble breathing, chest pain, shortness of breath, confusion, not arousable, then those are the indications to seek a medical attention immediately. Next. So what do you do if you do get sick? So the question is, do I have COVID-19? Is this the flu, the common cold, or allergies? So a test is needed to confirm this diagnosis because symptoms are overlapping. Mild to moderate symptoms can be treated at home. This includes cough, fever, some level of shortness of breath, fatigue, muscle aches, headache, runny nose, sore throat. A fair number of people describe loss of sense and smell, and 10% have diarrhea, but 17 to 19% have no symptoms at all. Next slide. When to go to the hospital. So I indicated this before. If you're short of breath and you can't manage it, you need to go to the hospital. Chest pain, chest pressure, bluish lips or face, confusion. If you're having difficulty arousing someone, they need to go to the hospital. Next slide. I just wanted to make one more comment about shortness of breath. Each individual has to gauge for themselves what qualifies an emergency. I think that that's something that as an emergency physician, everyone has a different level of emergency. So if you're short of breath, you need to seek attention. So there's two tests that we talk about. There's the RT-PCR, which is the reverse transcriptase PCR test, and then there's the antibody test, which checks for IgG and IgM. These are the main tests that are out there. The one that's available in most places is the PCR test, and that's the nasopharyngeal swab. Basically, you take a swab. Someone who's experienced doing it will take a swab and essentially put it back all the way into your nasopharynx. It goes in about 2 to 3 inches. So it is important that it's done correctly. It goes all the way back, and you can often feel very uncomfortable and tear from it. But it's not painful. It has to be done properly, but the important thing to realize that there are false positives. It's only 60 to 70% sensitive. And if you're negative and you have symptoms, you have to presume you still have the disease. The antibody test is essentially, it has 2 parts to it. It checks IgG and IgM. So what IgG is, when antibodies first mount, you get IgM. IgM is acute phase. So that means you have active disease at that time. IgG is what forms as you develop immunity. There are overlapping, there is an overlap between the time you have IgG and IgM. So, and as I said, there are a high rate of false negatives, and you can remain infectious. This is a very key point. The length of viral shedding is not clear and can persist as well in the feces for days, up to 30 days in some cases. This is something that's going to be published really soon, and very important key thing to keep in mind. How long we will have antibodies will remain unclear. Next slide. Okay, so these are, just want to review these things. The IgM denotes active infection. IgG reflects that you've had previous infection and you have immunity. And IgG rises later. So initially you're going to have a rise in IgM at about 5 to 14 days. And the IgG starts to rise a little bit later, around 14 to 21 days. So this is important to know, because when we start to test people, when they go back into the economy and into the workplace, how are we going to assess the safety of our employees? How are we going to determine who can go back and who and who needs to stay home further. And just another point, I just want to reiterate again about the fecal shedding of the virus can persist even after someone's recovered. Next slide. Before I go into this, I want to talk a little bit more. Could you go back to the previous? So I'm going to talk about going back to the workplace later. So we're going to go back into the tests, the PCR and the IgG and IgM antibody test. So it's understanding this will tell us how we can interpret each individual. Next. So what are our risk factors for getting sick? There's a 2 to 3% case fatality, but that's not actually, that may not be accurate, because we may have, once we do broad testing, we're going to see that there are a lot of asymptomatic people who've actually probably recovered. And once we have broad-based testing, that denominator is going to become more clear. So older individuals, people with heart disease, hypertension, chronic lung disease, diabetes, kidney disease, cancer, immunosuppression, HIV, AIDS, and skilled nursing facilities and homeless are very vulnerable as well. Next. So if you're at home and you think that you're sick, you don't have a test, you don't have access to a test, so you could be suspected, you may be confirmed, or you may be false negative. Things you can do at home are you can hydrate, Tylenol for fever. We are recommending avoidance of ibuprofen if possible, because there's some unclear, unconfirmed linkage of non-steroidal anti-inflammatories with worsening of disease. But so if you can avoid taking ibuprofen, it's better. Over-the-counter decongestants are good. Inhalers, you want to isolate from family and pets. If you are ill, separate bedroom and bathroom. Again, maintain telecommunication with your physician if possible. You want to maintain social distancing in the home. So if you're ill in your home, you should be wearing a mask to protect other people in your home. All members of the family, frequent hand washing, frequent disinfection of all commonly touched surfaces. Next slide. I just wanted to talk about a couple of things that haven't been talked about much, but proning. In the hospital, people that are being treated with supplemental oxygen or on ventilators, we've been proning them. What that means is we've been laying them on their stomach, and that recruits the basilar lung cells and basilar lung tissue to really expand that portion of your lungs, because there's a lot of collapse. So at home, if you're having some difficulty with breathing, you can use inhalers, and also you can lie on your stomach. So if you're surfing your phone or the internet, do it on your belly. Lying on your belly will really help you. If you can't do that, lying on different sides for as long as possible, and not lying on your back as much. People with lung disease, asthmatics, and COPDers should continue to use their inhalers. Pregnant women are at risk for preterm labor, premature rupture of membranes, things like that. So it's an extra, there's an extra consideration there as well. Next slide. I'm not going to go much into treatment. All these medications and treatments are under investigation. It's not really clear. Things that are more in favor right now are asterisks. It's not really hydroxychloroquine and azithromycin, but I still like them, so I starred them. But remdesivir, zinc, and convalescent plasma are the things that are really on the rise right now. And they have found that a lot of these individuals have been having small emboli, so blood thinners have also been used in the hospital setting. Next slide. So isolation criteria, I put a little asterisk next to this because I'm not sure I really fully agree. but this is what's out there right now on the CDC website. The WHO website's a little bit more stringent. So if your test is positive, when can you go back to work? I mean, we're all right now at home, but if you have to self-quarantine for at least 7 days since your symptoms first appeared. You can't have a fever, and so you have no fever for 3 days without the use of Tylenol type medications, and all your symptoms have to resolve. Or you have resolved symptoms and fever and 2 negative COVID tests. So essentially with these guidelines, you can go back to work, but as I mentioned, there is that it's not really clear how long people are infectious. So at a minimum, I would think that it should be 14 days or 21 days if you're 21 days even if you're COVID positive. But this is what's out there on the CDC website. If your test is negative, you can return to work when your symptoms have resolved and no fever, 24 hours without fever reducing medications. I would take this with a grain of salt, but this is what the recommendations are. Next. I do want to make a little plug for blood donation, because during a pandemic, blood supply is low, and having the diagnosis of COVID-19 is not an exclusion. The illness should be resolved for 28 days prior. Recovered people especially can make a great contribution with their plasma. Convalescent plasma basically means that you have antibodies, and you can share that with people who are critically ill and give them a chance. So contact your American Red Cross. Next. Returning to work safely. So this is what's on everybody's mind right now. And understand we're talking about this in a very, in a time frame that's rapidly approaching at this point, a week, 2 weeks, something like that. But I can tell you that there's nothing clear out there in terms of guidelines, but it depends on 3 things: the healthcare capacity, testing capacity, and certification capacity. So at the last time that I've been looking into this, it's been varying state to state. It may vary employer to employer. So everyone who's on here, everyone needs to really do their very best to protect themselves and their employers and their families, because when you go into work, you're exposing yourself and you're coming back home. And again, there's no standardization. It's a very fluid process. It's complex, and it has to be able to be modifiable. In the workplace, everyone needs to be wearing masks and social distancing. That's going to need to continue. Employers need to start considering modifying their workspace. So if they have different floors, spreading out the desks, different ways of moving around in an office space, because you also have to consider that different people in the office may have different levels of immunity. You have to also consider that maybe some people can continue working from home. Restroom hygiene, I want to bring this up. I haven't seen a lot out there for it, but I think it's really important. It's kind of concerning that with the fecal shedding of the virus, even after someone's recovered, every time a toilet is flushed, that you can have aerosolization of viral particles. And what that means is in the workplace, many of these toilets do not have lids. And so I think there has to be an introduction of some sort of practice in the workplace of how to address this. So I would suggest starting to consider that. Gloves, wipes, sanitizing of the bathroom, frequent hand hygiene, maybe aftermarket toilet lids. Everyone's an entrepreneur here, so that might be a good thing, but make sure you credit me. Next. Healthcare capacity. So it's very important also to understand that before we can go back to work, that each state must be over their current wave of COVID cases. They have to be past their peak, because if the hospitals are still overwhelmed, then we have to assume that there's going to be an increase in cases once everyone goes back to work. So the regional healthcare systems have to be able to support that, because there will be an inevitable second wave. Next. Finally, testing capacity. We're going to need millions of tests. And as we speak right now, the last I talked to, I spoke to Henry Schein, for example, yesterday. And there are currently, all the tests that are out there require a complex laboratory, meaning you can't, like a pregnancy test is pretty easy. You can go buy a pregnancy test in any drugstore. You pee on the stick and then immediately you see 1 or 2 lines. That's not where we're at with these tests at all. They need a lab to be processed. And so what that means is we're going to need millions of tests, and they're going to also require a hospital lab or separate lab. So that has to be factored in as well. From what I've been researching, I think the best way to kind of think about this is the two tests together, the PCR test and the antibody test, because when used together, they give us a lot of information. A positive PCR with a positive IgG and a positive IgM essentially will tell us that you likely have COVID-19 and that you have an active infection. Negative PCR and a positive IgG, in other words, will show that you have immunity to it, that you had it at one point and you're immune to it. So those are just 2 examples. So what we're going to want to determine when we start testing people, do they have active disease and do they have to go back home and isolate? Do they have immunity and they can go back into the economy, or have they never been exposed? So these are the things that I think are going to have to be very clearly delineated. Unfortunately, it doesn't look like the federal government is there yet in terms of guiding us. So we as entrepreneurs, business owners, and members of the community have to do our level best to educate ourselves and find out how we can get our employees tested and tested frequently. In Germany, for example, everyone has a certification that they maintain on their phone. And those are the people that are allowed into the workplace again. Next slide. And this is what I was just mentioning. Time-bound certification is necessary because we don't know yet what the length of immunity is. I think SARS had an immunity that lasted about 3 years. And we also have to be able to have widespread PPE for returning workers. I'm not talking about healthcare workers. I'm talking about appropriate PPE for people in the workplace, masks, those types of things, wipes. Next. So I want to thank you all. I hope I didn't take too much time, and I appreciate it. Thank you, Jeff.
Thanks, Deepa. It's great information to have. Hi, everyone. This is Jeff Banks. I'm the CEO at Nuventra. We're a drug development consulting firm. If we can go to the next slide, please. So I'm going to do a quick drug development primer to sort of set the stage to what I'm seeing with the drug development spectrum for COVID-19, SARS-CoV-2. So if you look at this sort of spectrum of drug development here, we typically start in drug discovery. We have to find drugs or molecules and try to test them against different types of diseases. And generally there's thousands of types of molecules you might test. Some of those progress into preclinical studies here. And unfortunately, this involves animal studies. It's just the nature of how we have to do things. We have to understand the toxicological profile of these drugs before we go into humans. So this can take quite a bit of time. It could be 3 to 6 years from discovery to completing all the necessary preclinical studies. Once that's completed, you'll see in the middle there, it says IND submitted. That's an investigational new drug application that gets submitted to the FDA or other regulatory bodies and basically says, we think we're ready to go into clinical studies and test the drug in humans. And if that's allowed to go forward, there's 3 phases of drug development. Phase 1 is typically healthy volunteers, trying to understand the safety of the drug in humans, trying to understand how the drug behaves in humans, understand the dose, what's an appropriate dose. Moving into phase 2 is when you start to get into patients, and this is more of a proof of concept to try to understand, does the drug work at various levels in patients? And then phase 3 is really sort of your pivotal controlled studies to make sure in a larger patient population you think it's doing what it might. And these can take thousands of volunteers across this, these 3 phases of clinical research, anywhere from 5 to 10 years even to complete these. But once you do complete everything and you think you have the full data package, you go back to the FDA with what's called an NDA, or New Drug Application. The FDA will review that, and if they agree, they'll approve the drug to go into the marketplace. And then there's what's called post-marketing surveillance, which is sort of evaluating in a larger population, is the drug behaving as you had anticipated. So this can take anywhere from 10 to 15 years. But what I'm seeing and what I'll show you today is just the unprecedented speed from my perspective that I'm seeing drug development for the SARS-CoV-2. So next slide, please. So it really is just an all hands on deck approach right now. And this is, I can only give some examples. I mean, this is changing day to day, but for one example is NIH has started this program. It's called the Accelerating COVID-19 Therapeutic Interventions and Vaccines, or ACTIV. And it's a collaboration between NIH, Centers for Disease Control, the FDA, the European Medicines Agency is essentially the European equivalent of the FDA, and then 16 global major pharmaceutical firms all collaborating on finding therapeutics for COVID-19. We're seeing just a tremendous effort in trying to repurpose current grant dollars from existing research to COVID-19 research. We're seeing pharma companies across the board redeploy full R&D teams to COVID-19, putting other research activities on hold, trying to help fight this terrible disease. And we're also seeing what we're doing is we're learning from past global health emergencies. We've heard a little bit today around, the SARS and the MERS, which are similar type of coronaviruses as COVID-19. If you remember the swine flu in 2009, and actually quite a bit recently from the Ebola outbreak, we've learned some things from that we're applying today. Certainly HIV has been with us for 40 years. That's also another RNA retrovirus that we've learned quite a bit from as well that we're applying right now. So next slide, please. Similarly, at the FDA, they started a program called the Coronavirus Treatment Acceleration Program. As I put that sort of spectrum of drug development up on the first slide, you're interacting with the FDA along the entire path there. It's not just at the IND and the NDA stage. You're interacting with the FDA, you're going to meetings, you're getting their feedback and their input. What we're seeing now is they have a team throughout the agency that is being deployed with just ultra rapid input for drug developers to gain their insight on their programs. Response times, they're saying, are within hours. And typically that might be months. And so just a tremendous level of effort and support on behalf of the regulatory agencies to support drug development right now. So some of the metrics they put on their website is, as of April 16th, the FDA received 950 inquiries and proposals related to COVID-19 drug development. 950 separate entities or individuals, maybe a physician or a pharma company or a group of people submitting requests for support from the agency. They're reporting 72 active trials of therapeutics that they're aware of as of the 19th, and another 211 drug programs in the planning stages. But this is really sort of the tip of the iceberg. If we go to the next page, sort of a clearinghouse of information for clinical trials across the world is this website called clinicaltrials.gov. And it's actually mostly required now when you start a clinical study. If you're going to publish it, you have to have your study listed on clinicaltrials.gov. And April 20th, I did a search for a keyword, just COVID, and it popped up 727 clinical studies. And this is since December. I mean, the level of studies and information that's being generated is, again, I'll use this a lot, is really just unprecedented. And it's not just therapeutics and vaccines. I mean, we have to understand the sort of disease progression, the infections rate, the etiology of the disease. So a lot of observational studies are ongoing as well. There's a couple other sites. It's called medRxiv and bioRxiv. This is sort of a clearinghouse as well for laboratory and health science disease research. And I put on here, it's called a preprint article. So when you do sort of research in this respect, you write up your results in a publication and you submit that to medical and research journals in sort of a peer-reviewed framework. So it has to go out, has to be reviewed by other peers in your field, and that can take some time. What these websites are doing is they're taking those articles and basically publishing them right away as they're also going through peer reviewed. So that you have to take it with that sort of grain of salt, if you will. They haven't been vetted by the scientific community, but all their methods are there, all their information is there. And so, sorry, something just popped up on my screen. And so what I'm seeing, similarly as of April 20th, there's almost 2,000 research articles that have been posted there for COVID-19, SARS-CoV-2. I mean, just incredible amount of effort. And as Giles mentioned, and it's just really a global effort across the board of collaboration that we're seeing right now. So next slide, please. So this is a common question people ask is, when will a vaccine be ready? My answer to that is really not just a vaccine effort. This is the kitchen sink sort of approach to drug development. We are throwing absolutely everything we can at this. And vaccines, from my perspective, typically are to prevent types of symptoms. And they're very specific and, typically very specific for the pathogen you're looking at. So it does take time to develop them. But there are therapeutics and there are drugs that are currently on the market or available for other diseases that could be used right now to help treat patients. And we're seeing that happening. And obviously, as Deepa talked about, they're looking at different types of drugs that are currently in the pharmacy to treat COVID-19 symptoms, like, how can we inhibit the viral replication? How can we treat the very damaging lung injury that they're seeing? And really this sort of hyperimmune response that seems to be the underlying sort of etiology of this pathogen is what's called cytokine storm. So there are multiple drugs out there that could address that may not have been, quote unquote, tagged for COVID-19 before, but we're looking at repurposing them as quickly as possible. So next slide, please. So again, it's not just a vaccine effort, but interestingly, as I tell people, and I'll show you an example here in a little bit, there are multiple vaccine candidates that have already been identified. They're already working towards trying to get those available to people, and I'll talk about that. There are numerous existing drugs on the pharmacy shelf right now that are being repurposed. There's numerous, quote unquote, new drugs in development. So these are drugs that have possibly already gone through the non-clinical studies that I talked about, or have actually fairly good clinical data already from clinical studies in development. They just haven't yet been approved by the FDA or the EMA or other regulatory bodies. And so we're looking at repurposing them as well. But the underlying theme here is that we all have to test them in controlled clinical studies for COVID-19. The last thing you want to do is you want to have a drug that you rush through and it actually kills more people than COVID-19 would. And so we have to make sure first that it's safe, that it actually works as designed. We understand how the drugs behave in the body, what's the dose, what's the proper dosing to give, how are the drugs cleared. All of that is what comes into a typical drug development program over that 10 to 15 year span that I mentioned, is now being condensed in sort of rapid time. We're not cutting corners. We're not doing that, but we're doing everything we can, from my perspective, that I'm seeing with the drug developers, to move this along as fast as possible. So next slide, please. So as I said at the beginning, there's just so many, and it changes daily, types of therapies that we're investigating, from the antiviral therapies, and I'll talk a little bit about those, cellular therapies, the immune therapies, which are vaccines, anti-inflammatory drugs, repurposing anti-cancer drugs, anti-cytokine drugs. I don't know if many of you remember the drug thalidomide was a drug that back in the 50s caused horrible birth defects in women who were pregnant. It actually is on the market today as an anti-cancer. Certainly you're not going to give it to patients who are pregnant or could become pregnant, but that's even being tested for potential COVID-19 antiviral activity. Inhaled nitric oxide has been tested in a study for years. And there's some evidence that it might help in acute respiratory distress syndrome, or ARDS, which is sort of the underlying condition that we're seeing as well, presented with COVID-19 patients. So just across the board, understanding what's there. And then of course, the chloroquine, hydrochloroquine. I'm going to talk about that as sort of an example of why we need to move fast, but also why we need to be cautious. And so I'll show you some actual data on that. So next slide, please. So here's a great example of what I'm seeing out there. And this is one investigation that was posted on the bioRxiv. So this is a preprint, it hasn't been peer reviewed, but essentially this group took 12,000 drugs and they put it through what's called a high throughput assay. And this is a typical type of assay that's done in that drug discovery phase that I mentioned, where you're basically throwing a lot of drugs at an issue to see if something works. And what they found is there were 27 drugs that were effective at halting the viral replication, basically in a petri dish. And that's where the chloroquine, hydrochloroquine thing started, is we found that there was some inhibition of viral replication. I'll talk about that again, as I mentioned a little bit later. But there were 27 drugs that were found to halt viral replication. 17 of these already have extensive safety data from clinical studies in non-COVID-19 diseases. So they have a leg up. We have an avenue potentially to move these faster along. 4 of these drugs have already been FDA approved for other indications. So the FDA has said they're safe enough and they're efficacious enough in these other indications. So the question is, can we test them in COVID-19? And then critically, it's also, are they actually going to work at safe dose levels? And so they determined that 6 of these drugs could potentially be safe and efficacious at doses in humans that make sense. So all of this, again, is one article, but you can see from here that in these drugs were purposed for rheumatoid arthritis, Crohn's disease, osteoporosis, cancer. We have the means and the technology to really advance this very quickly. The lagging issue here is it takes time to test these in humans because we just don't want, obviously, to have safety issues down the road. So next slide, please. So the antiviral therapies, remdesivir from Gilead as a repurposed drug. Again, it showed in vitro and in vivo antiviral activity against MERS and SARS. There's some preliminary antiviral activity in vitro, which is basically a petri dish. A phase 2 study in 2,400 patients is ongoing, but the initial readouts are positive. So there's a lot of excitement there. There's a lot of anticipation, but we still need to conduct the clinical studies to make sure that they're effective. And there's other ones here as well for influenza, Hep C. I put this one on for HIV, and then I was reading some information last night that actually the NIH is recommending against this treatment for COVID because of negative clinical trial data and what's called pharmacodynamics, or how the body is responding to the drug. So that's just rapid information that's being available. So again, here's a vaccine example. And typically when we look at vaccine development, we target what I like to call the weakness in the pathogen. And the weakness is per se what the pathogen can't do without. It has to have certain aspects of its genetic makeup or protein makeup that allow it to be as bad as it is, if you will. So we have sort of a schematic over here, which is a simple RNA virus that has a genome that is 30,000 base pairs. And the genome, as for comparison, for a human cell is 3 billion base pairs. So it's a fairly simple pathogen. Obviously, it has the characteristics that are causing this damage in lung tissue and patients. And so what the vaccine effort that I'm seeing right now is they're targeting what's called the spike protein, sort of circled there on the surface. And the spike protein is just a combination of amino acids that make it such that it can interact with a human cell. The human cell is being used as a factory, basically, by the virus. It injects, the spike protein attaches, the RNA genome is inserted, and it hijacks the human infrastructure to make all the replicate viruses, and then that's how the virus spreads and moves on. And so the theory here is, if we can mimic the spike protein separately and expose the body to that, then we'll elicit an immune response, essentially developing antibodies that hopefully will be more ready to go if the body doesn't come in contact with the actual virus down the road. So neutralizing antibodies against the spike protein, which we believe is something that the virus can't do without, or it won't be as pathogenic, basically. So next slide. So here's an example of just sort of how fast, from my perspective, things are going. So Moderna Therapeutics is developing a vaccine right now. January 11th, the genetic sequence for SARS-CoV-2, their genome, the 30,000 base pairs, was made available. Within 2 days, by January 13th, we had a vaccine candidate. They had a vaccine candidate. They've essentially identified the vaccine they think is appropriate. By February 21st, within little over a month, they filed their investigational new drug application. And if you remember on my first slide, that can take anywhere from 3 to 6 years. The FDA actually reviewed and allowed the clinical study to proceed in the first dose of a healthy volunteer for their drug, which is called a messenger RNA drug. And they have a particular type of platform, if you will, that has been tested in the MERS and the SARS viruses as well. So they had a leg up here, but this is a very specific entity that they've created against COVID-19. 63 days from essentially drug discovery to human first dosing is just unprecedented. And so they're now testing this in an open label healthy volunteer study, and they have plans for phase 2, phase 3 studies in 2020. So again, normal vaccine development might take 10 years. For Moderna, it could be 12 to 18 months, and even sooner for emergency access or participating in the clinical trials in these phase 2, phase 3 studies for this drug, for this vaccine. Next slide, please. So just real quickly, cellular and immune therapy. The cellular therapy, there's these things that we're seeing tested in natural killer cells that help target viruses or even stem cells. Immunotherapy, as Deepa mentioned, the convalescent plasma has been shown to be effective in influenza A and the Ebola outbreak. And then, of course, the neutralizing monoclonal antibodies. There are other companies doing this as well. This other company, Innovo, has a DNA-based vaccine essentially looking at the spike protein as well. So we don't know if these vaccines obviously are going to work. The theoretical science is there. We understand that, what the genetic makeup is of the pathogen. We have what seems to be a reasonable target. Now we have to make sure we test it in clinical studies. So next slide, please. So again, I touched on the chloroquine, hydroxychloroquine is sort of an example of how to be cautious with this, but also have obviously hope. I mean, overall, the amount of research we're seeing is just tremendous. And so there certainly is hope. And I'll talk about sort of my perspective of what I see the availability of a therapeutic treatment and vaccine for COVID-19 in just a moment. But so if we look at hydroxychloroquine and chloroquine, there was this study that was done in France with a small number of patients, as you can see here. It was recently published, again, a preprint that was made available. And if you look at the graph here, it looks quite good. On over 6 days of dosing on the x-axis versus the essentially viral load, percent viral load of COVID-19 on the y-axis. You can see in green there, for 6 patients that were dosed with hydroxychloroquine and azithromycin, the viral load went down to essentially 0. For hydroxychloroquine only, it was a little bit higher, and then for the control patients, it was higher, suggesting that there might be some potential here. And this is on the back end of what we determined was some in vitro activity of these drugs against the SARS-CoV-2 pathogen. So promising data, but small. An open label means that the patients knew what they were getting. Still, there's an objective measure of a PCR-generated viral load assay that it's hard for there to be a placebo response with that. So this is promising. But if we move to the next slide, this is the cautionary tale here is another article that was made available as a preprint, again, peer-reviewed, was a retrospective analysis of 368 COVID-19 patients in the VA medical system. Now, this is a review of medical records. So these patients were not prospectively enrolled in a clinical study. We didn't define what dose we were going to give them. We looked back and said, what happened to these patients when they were given these drugs? And some of the things that stood out were the death rate that they identified for this. As you note here, hydroxychloroquine alone was more than double that for the control group. And hydroxychloroquine plus azithromycin was still higher than the control group. And when you looked at the risk of mechanical ventilation, it didn't seem to be any benefit to adding the 2 drugs above. And this again is a retrospective, not a randomized controlled study, but it's a data point that we have to consider. You have to understand that there's the efficacy component and the safety component. And so there's some questions about this right now, and this is just being made available. But it was interesting, the author's conclusions said, this is word for word, these findings highlight the importance of awaiting the results of ongoing prospective randomized controlled studies before widespread adoption of these drugs. And that is true. There's by no means we should stop development and evaluation of these drugs. We just need to do it cautiously before we provide broad-based recommendations as to how they should be used. So the final slide here is sort of what I see over the next 12 months. And so I think what we're going to continue to see is, as Deepa mentioned, is physicians on the front line are trying what they can with what's called off-label use of drugs right now. There's no drug approved for COVID-19. So they're looking at what's available. They're researching the literature. They're trying to use different things, antivirals that are available and whatnot. I think that's certainly going to continue. We're going to see widespread clinical trial access to vaccines, existing drugs, and new drugs. And typically in a clinical study, you have this criteria in order to get in. And the criteria that I'm seeing for patients to get access into these clinical studies is very, very small. It's basically, do you have COVID-19? And that's pretty much it. So there's going to be quite a bit of access for patients who have COVID-19 to the drugs that are currently in development under clinical studies. And we're going to continue this kitchen sink approach. I don't think this is going to be slowing down. The broad-based global collaboration, really unprecedented from my experience that I'm seeing. We'll start to see emergency use, compassion use, authorizations. This is within the power of the FDA and EMA to do that. And that essentially means outside of a clinical study, not approving a drug, still allowing patients to have access to it. So we're really throwing what we can at this to try to help this terrible, terrible disease. And I do believe that given what I'm seeing, we're going to see an approved drug by the end of this year, early in 2021, whether that's a vaccine, a treatment. They have to go through a similar type of path to some degree. And there's an ongoing updated information at the NIH here with a link here. And finally, just really quickly, for me, it's interesting. Imagine such an effort if we're to be placed on other diseases. There's another retrovirus out there, RNA virus, infectious disease, that's been with us for 40 years, that in 2018 killed almost 700,000 people globally. That's HIV. Can you imagine the type of damage we could do to that if we had a similar type of effort that we're seeing right now for COVID-19? So next slide, and I think we're in Q&A.
Awesome. Jeff, Deepa, Giles, this is amazing. Just have a few questions as a reminder to those on the call. If you have a question you'd like to ask, if you can raise your hand, we're just gonna have time for a few, or throw it in the QA panel. Start with Matthew Wolf from Detroit. Can you talk a little bit about Sweden's herd mentality and what you think of it, please? And I'm not sure who is best to answer that, but I'll let you all decide.
Deepa, you want to take that, or Giles?
Sure. I think that's, for me, more of an ethical question, because essentially, for those who don't know, Sweden took a stance that they allowed large people to gather. So their curve didn't flatten, it went up, it went straight up. So they had a high number of deaths. And their argument was that they would develop herd immunity, and they think that they're going to get it by May. I think that's more of an ethical issue, and it spread, I think, quicker through nursing homes, and they didn't try to contain it. Meanwhile, the neighboring countries, I think if it was Norway and Denmark, shut their borders down and had a very different response. So, I mean, it comes down to whether you think, every life's important.
All right, just informal poll. For the three of you, when will you be comfortable hopping on an airplane and going somewhere with your family?
Question. I had travel planned earlier this year to Asia. Obviously, that got pushed back. Initially, we were pushed pushing it back till May, but I think I wouldn't take a chance till probably the summer, and June at the earliest to really get on a plane to go anywhere. Just because in a plane you're in a confined space, and you don't know what's around, and we've seen that there's been cases spread. I think honestly, I wouldn't take my family until we've got vaccines and real control over the virus. But if I had urgent business travel, I would definitely take precautions and masks, and maybe mid to late summer. That's my view.
It's a wait and see for me right now.
I traveled twice to New York in the last couple of weeks, and it's very disconcerting. The airports are empty, for sure, but there's just too much to think of between hotels, airports, rental cars. It just, you feel extremely vulnerable, and it's just not a good situation. I mean, I don't see anything earlier than July, but I mean, I think it's a very fluid process, and I would probably wait. I mean, I think that I'm very optimistic about, after Jeff's presentation and what I've been hearing about vaccines. So, but I think that we're in an unprecedented time, and it's going to, things are going to resurge. So it's hard to know. And our kids have to go back to school, and all of that is going to add to it. So, I mean, it's not just vacations, kids traveling across the country to go back to college. I mean, that's like a vacation, right? I mean, it's still traveling and going somewhere and doing all that stuff. I mean, there's so many similar types of scenarios that isn't just about a vacation. And a vacation, you're there with a family, so you can kind of manage what everyone's doing. But those of us who have older kids and they have to go back to school and it's not local, I mean, there's just, I think that has to happen first before we can talk about vacations. Well, actually, I should say vacations are probably easier to manage than all those other situations.
The toilets, I didn't think about that. And on airplanes, that's certainly, that was eye-opening for me when you talked about that, Deepa. That made me very nervous about flying.
Well, even not only flying, but in your office space, a lot of these offices, we only have toilet lids in our home, in our homes really, but in the public restrooms, if you're in an office the toilets have very strong flushes, and kind of disgusting if you think about it, but they have to kind of really consider how to manage that. And as an employer, I don't think any state's gonna, or the federal government's gonna come up with a plan. So you have to really consider how in your own workplace are you going to manage, that type of thing. When I worked in the hospital a couple of weeks ago, there were wipes in the break room bathroom, and went in there with a mask, went in there with I wasn't wearing gloves, but I wiped everything down, and then when I left, it was just like, oh my gosh, a lot to think about.
Hopefully someone on this call or someone in EO is inventing toilet seat covers for... Whoever
does that has to include me, because I texted someone last night. I said, we need to do that.
It seems like we could solve for this. Giles, you've got the import-export down. Why can we not solve for that in like the next 10 minutes and get this figured out and done?
Seriously, because just on every... it'll be good to go forever.
Well, thank you so much. This content was incredible. For those who stayed with us, really appreciate it. The recording will be at eoraleigh.com/coronavirus. The deck will be there. Giles, Jeff, Deepa, thank you so much. Really appreciate your time today. Thanks for sharing as much as you did and working so hard to put this presentation together. This was really, really impressive. Thank you.
Thank you.
Thank
All
you.
right, stay safe. Take care.